My name is Nathan Tucker, instructor in medicine at Massachusetts General Hospital and the Broad Institute of MIT and Harvard. The work that I'm presenting today is entitled A Single Cell Analysis of nearly 300,000 individual cells from a non-failing human heart. So to explain a little bit what that means, we're interested in RNA biology, so expression profiles of output of the genes within the heart. So in the past, usually what folks have done is treat heart as a tissue, take a bulk core of tissue, which is a mixture of many different cell types that might be present. You end up with an average expression value for that tissue that you're going to measure. So a lot of really great work's been done in both in health and disease biology. We can understand what's different between left and right side, upper and lower chambers. We can get some understanding of looking at disease versus health. But one big problem with that is, there are many ways from which these differences can arise. So our goal is to kind of deconvolute something that's a tissue, we call it heart, into actually its cellular components. What we did for this work, we worked with collaborators at the University of Pennsylvania that had non-failing donor tissue that was treated as for transplant. So probably as close as you can get to a biopsy of a sample as we can do. We take that into the lab. We isolate actually single nuclei rather than cells so that we can take frozen tissue. Use a microfluidic device to separate these into individual cells and profile transcriptomes of each one of these individual cells to a given level. So at that point, we let the data kind of drive our discovery and look at, on average, about 1,500 transcripts per cell in the nucleus, and identify what the different cell types might be across those four chambers. So major findings would be, 9 major cell classes that I think most of the folks watching would be familiar with. So, cardiomyocytes, vascular-associated cells, and immune component as well. But you can get a little more granular than that too. So are there different types of cardiomyocytes? Are there different flavors of fibroblasts and the like? So it ends up being about 20. We're pretty conservative with about 20 subclasses of cells that we can confidently talk about. So what you end up getting out of that is probably the best description from a transcriptional standpoint of these individual cells that has been presented to date. So there's been a lot of cardiomyocyte work, and I think people have a good handle on their expression profile. But these other cell types have been a little bit of a black box. So this is a well-powered study of non-failing donor tissue to get a handle on what these other cells look like from a transcriptional standpoint. This is a really large technological and analytic challenge. So the data are huge. The pipeline to get the tissues into the lab that we've established has been an endeavor. Understanding how to work with the data has taken us a year and a half since we started. So we'll hope to provide this kind of framework to the folks who are similar needs. We'll be going into the disease versus normal comparison, heart and other tissues as well, so hopefully inform what those not only samples are like heart, as a tissue, but cardiomyocytes act this way, or fibroblasts are acting this way, or the populations change based on disease stage and the like. So that's the next immediate step. There'll also be technological development as in any of these endeavors as well. So other things we can pull out, I can provide the best current description of sidedness or cell composition for folks who want to take these findings into the lab and to model what they're working on in real context. We're also pushing into identifying potential drug targets, whether it's repurposing of drugs that have been approved or things that are deemed highly druggable just due to like the protein sublocalization. You can say these given genes seem to be expressed specifically in the cell type that hopefully inform the way that you're going to model your drug's function. So we're really excited about the work. I think we're going to see a lot in this vein going forward. We're part of a session where the entire thing is all single-cell RNA sequencing. I think we're by far the most scaled. We might be the only one that's in human tissue, but I would say these should be data that anybody who's working in the lab or thinking about drug development should consider in their process.